Hey everyone, this is Sir Amel, and in today's video I will be discussing with you on the topic glycogenesis. When we eat our food, the food will be broken down by our digestive system and associated enzymes. The complex organic molecule will be broken down into simpler organic compounds that will be utilized by the cell's body. Okay? These nutrients are essential for the proper functioning of the cell's metabolic activity, and glucose is one of these essential nutrients that we get by the breakdown of food. Glucose is a very important monosaccharide that will be utilized by the cell of the organism to make energy. When several glucose molecules are being linked together, they form a more complex compound which we call as glycogen. So glycogenesis, as the name suggests, genesis means to form or to synthesize or to make. Okay, so glycogenesis is the process of synthesizing glycogen or the process of formation of glycogen. And glycogen is a polysaccharide which is a polymer that is made up of many monomeric units of alpha D glucose. Glycogen is the storage form of glucose in animals, as is starch in plants. Glycogen also provides an important energy reserve for the body. The two main storage sites of glycogen are the liver and the skeletal muscles. The glycogen is also stored as granules in the cytosol, and these granules contain not only the glycogen but also the enzymes and all the regulatory proteins that are required for glycogen metabolism. And in this particular video, we'll be talking about how glycogen is made. Alright, let's get into it. In the first step, we have a molecule of glucose. Okay, this molecule of glucose can be anywhere, well, not precisely anywhere, but more specifically in the muscles and in the liver. So glucose molecules in the liver and in the muscles will be utilized to make or to synthesize glycogen. So there are two enzymes. One is called hexokinase, and this particular enzyme will convert glucose into glucose 6-phosphate. Okay, and that happens in the muscle cells, but we are mostly concerned with the enzyme that will convert glucose into glucose 6-phosphate in the liver, and that is glucokinase. So glucokinase will convert a molecule of glucose into glucose 6-phosphate in the liver. Okay, so here what will happen is energy will be required. Okay, ATP will be required, a molecule of ATP will be required. Glucokinase will take one of the phosphates from ATP and transfer one of the phosphates into the carbon number 6 hydroxyl group of carbon number 6 glucose molecule, and this will form glucose 6-phosphate, and a molecule of ADP will be liberated. So that is the first step, conversion of glucose into glucose 6-phosphate either by hexokinase in the muscles or glucokinase in the liver. The next step of glycogen synthesis is the conversion of glucose 6-phosphate into glucose 1-phosphate by the enzyme phosphoglucomutase. Now in this step there are actually two reactions. The first reaction is when the serine residue of phosphoglucomutase with its own phosphate group will donate it to the hydroxyl group of carbon number 1 of this particular glucose 6-phosphate to form glucose 1,6-bis phosphate. Okay, and in the second step, what will happen, that same serine residue of phosphoglucomutase will take up the phosphate group of carbon number 6 of this glucose 1,6 bisphosphate, and then we should be left with glucose 1-phosphate. In the next step of glycogen synthesis, glucose 1-phosphate will be converted into a sugar nucleoside which is called as uridine diphosphate glucose, and in this step we have glucose 1-phosphate will be condensed with a molecule of UTP or uridine triphosphate, and in this reaction the negatively charged oxygen of this glucose 1-phosphate will attach to the first phosphate group of UTP to form UDPG, and also liberating pyrophosphate. And this particular step or reaction is catalyzed by the enzyme UDP glucose pyrophosphorylase. So in this step, two molecules of phosphate will be liberated from UTP, okay, and what is left, we have the glucose 1-phosphate here. The glucose 1-phosphate will be condensed with what is left, that is UMP, okay, and uridine monophosphate from UTP. So whatever is left from UTP, that will be condensed with the glucose 1-phosphate, and what do we get? UDPG or uridine diphosphate glucose. In the next step of glycogen synthesis, we have the initiation of the formation of a glycogen primer. But in the absence of a glycogen primer, a specific protein will be used, and this is known as glycogenin. This particular protein will have a tyrosine residue, okay, and the hydroxyl group of this tyrosine residue of glycogenin will be attached to the carbon number 1 of UDPG to liberate UDP. Okay, so UDP will be liberated, and we have now a glycogenin glucose molecule. And now this glycogenin glucose molecule that is being formed will be attached to another UDPG, another molecule of uridine diphosphate glucose at the 4th carbon atom, at the hydroxyl group of the 4th carbon atom, and again another UDP will be liberated, and you get this glycogen primer. So you have the glycogenin protein here, so we have this glycogenin glucose primer, okay, and when this is formed, we liberate, or rather a molecule of UDP is liberated from UDPG, and when another UDPG molecule is being added to the hydroxyl group of carbon number 4, another UDP will be liberated, okay, and this process will repeat itself again and again till we get a glycogen primer. So you have here a glycogen primer, okay, and so you have this glycogen primer that we can see here, and again, like I said, this reaction of addition of glucose by UDPG will proceed, or rather repeat itself, for at least 6 to 7 times till we get a very long chain, or 6 or 7 glucose residues that is being added to this glycogen primer. And in the next step, the enzyme glycogen synthetase will take over, and new glucose residues will be added along the chain, at the non-reducing end of the chain. Okay, so glycogen synthesis will make these alpha 1,4 linkages, that is a bond between carbon number 1 and carbon number 4 will be formed of each glucose molecule. Next step of this elongation and the formation of glycogen is the formation of branch points. And how will this happen? This will happen through the enzymatic activity of an enzyme which is known as glycosyl alpha 4,6 transferase. This particular enzyme will take some of these glucose residues from the main chain, 5 to 6 or even 7 glucose residues, or even 8 for that matter, and transfer them to carbon number 6 of the first glucose molecule of the glycogen primer. Okay. And then we have a branch that is formed, and that is the alpha 1,6 branch. So we can see here. Okay, so those glucose residues are now being transferred to this alpha 1,6 branch point. Okay, and again new glucose residues will be added at this new non-reducing end that is being formed again by glycogen synthesis, while in the old non-reducing end more and more glucose residue will be added again and again by glycogen synthesis. And this is how glycogen synthesis is done. Okay, and this process of elongation will go on and on and on again and again till we get this huge polymer of glucose molecules, which is called glycogen. This will be a molecule of just a simple illustration of a glycogen molecule. Alright, so we can see here that in this particular picture we have the glycogen primer here, which is formed by glycogenin, or rather which is initiated by the protein glycogenin. Okay, the red color represents the primer that is being formed by the action of glycogenin and also the release of glucose from several uridine diphosphate glucose. Okay, and then you can see here the dark purple color. These are the branch points that are being formed at the main chain. Okay, that is transferred by the activity of the enzyme glycosyl 4,6 transferase. And again at these branch points more branch points will be formed, and the chains keep on elongated by the glycogen synthesis activity. All right, and we can see that in each of these chains that are being formed or being newly synthesized in the glycogen, each chain will have about 12 to 14 glucose residues. Hope this video has given you a clear understanding of the process and steps involved in the formation of glycogen. Don't forget to like this video and subscribe to my channel. Thank you.